I can't believe we're here. Molecular Genetics, Chapter 12. There are 4 sections: DNA, the genetic material, replication of DNA, DNA, RNA and protein, and gene regulation and mutation. So DNA is the genetic material that contains a code for proteins. The main ideas from your 4 sections: the discovery that DNA is the genetic code involved many experiments. DNA replicates by making a strand that is complementary to each original strand. DNA codes for RNA, which guides protein synthesis. And gene expression is regulated by the cell, and mutations can affect this expression. Here are your essential questions. And your new and old and continued vocabulary. So section 1, DNA, the genetic material. The discovery that DNA is the genetic code involved in many experiments. So after the rediscovery of Mendel's work, scientists began to look for the molecule involved in inheritance. For many years, scientists struggled to determine if DNA or protein was the source of genetic information. That was the big question. And so this guy named Griffith comes along and does this experiment where he was searching for genetic material. It involved in transformation between 2 forms of S. pneumoniae. Set the stage for search to identify the transforming substance. Now you have a picture of this in your book. I highly suggest you go take a look at it because this is on your test. Avery identified the molecule that transformed the R strand of S. pneumoniae into the S strand. He concluded that when the S cells were killed, DNA was released. Our bacteria incorporated this DNA into their cells, thus changing into S cells. So, discovery of the genetic material by Hershey and Chase. They used radioactive labeling to track the bacteriophage, DNA, and protein. They concluded that the bacteriophage DNA was injected into the cell and provided the genetic information needed to produce new viruses.
It was already known that when bacteriophage infects bacteria, the bacteriophage replicates bacteria. So the question that they asked was, what is the genetic material of the phage? Is it the DNA or the protein? And to answer this question, they designed an experiment in which the code protein of the phage was labeled with S35 and the DNA was labeled with P32. The bacteriophage was then allowed to infect the bacteria. Once the infection was over, they used a blender to detach the bacteriophage that were attached to the bacterial cells. The mixture was then centrifuged and checked for the presence of radioactivity. It was seen that in the second set, the bacterial pellets showed very high radioactivity. This radioactivity was due to the presence of P32 present in the DNA. Hence, based on this, it was concluded that the DNA is the genetic material.
First of all, DNA is a type of nucleic acid. And if you remember the biomolecules, remember how the biomolecules included carbs, lipids, proteins, and nucleic acids. Nucleic acids are the type of biomolecule that DNA falls under. And like all biomolecules, there are building blocks called monomers. The building block of a nucleic acid is a nucleotide. Okay, so I know that I'm kind of bombarding you with vocabulary, but it's really important to understand the parts of a nucleotide because otherwise you don't know what DNA is made of. And if you don't understand what DNA is made of, you won't understand how it functions. And if you don't understand how it functions, well, that's going to be really difficult when we really start understanding how we work. Before I get into the 3 parts of a nucleotide, I do want to point out the name of DNA. So DNA stands for deoxyribonucleic acid. That's what the Na part is, nucleic acid. And that D, that deoxyribo, That's for deoxyribose, which is a sugar. So nucleotides have 3 parts, and one of them is a sugar called deoxyribose. The next thing is a phosphate. The phosphate is involved with the sugar in helping hold the DNA structure together. We sometimes call DNA as having a sugar phosphate backbone. But the most important part of the nucleotide is the base, because the bases actually code for traits. So as far as the bases go, again, those are the important parts that control your traits. They really directly code for your traits. There are 4 bases in DNA.
So what Amoeba Sisters is getting ready to talk to you about is Chargaff's rule. C equals G and T equals A. So C's and G's will always match up, and T's and A's are always going to match up. You will never see a G and an A together. You will never see a C and a T together. Think of round peg, square hole.
A, T, G, and C. The A is for adenine, the T is for thymine, the G is for guanine, and the C is for cytosine. There's a really nice little saying that can help you remember which one goes with which, because these bases actually pair in a correct pattern. And if you don't have them paired correctly, or maybe they get mismatched, that's actually what we call a mutation, which we'll get to a little bit later. But here's the little way you can remember it. Apples on the tree. That tells you that A for apples, T for tree, A and T always go together. And then the other little verse is cars in the garage. That can help you remember that the base C always goes with the base G. And so you kind of know how they match. And DNA pairs this way. DNA has 2 strands. So there's nucleotides running up one side, and there's nucleotides running up the other side. The base.
You will need to be familiar with the scientists that were part of discovering and designing the structure of DNA.
Bases are what pair in the middle. They're usually held together by hydrogen bonds. The DNA is also twisted in something we call a double helix shape. And so as the strands of the DNA is twisted around, the bases should be all in the center. The sugar and phosphate make up the sides. So let's review what we talked about today. We talked about the importance of DNA.
You need to know about the top rail and the bottom strand. So on the top rail, the strand is said to be oriented 5 to 3, and the strand on the bottom runs in the opposite direction and is oriented 3 to 5. So as you can see here, we have 3 to 5, and we have 3 to 5. And then it matches up. So we have A to T, C to G, T to A, G to C. In prokaryotes, it's worth noting that DNA molecules are contained in the cytoplasm and consist mainly of a ring of DNA and associated proteins. However, in eukaryotes, the new ones, us, it's organized into individual chromosomes and it's held in the nucleus. So this is the chromosome structure that you will need to be familiar with. To fit into a cell, DNA coils around a group of bead-like proteins called histones. DNA plus histones form something called a nucleosome, which group together into chromatin fibers that supercoil to form an end in a chromosome. So here we are at the beginning, going through the process, being knitted together, and there we go.